Hi, I'm Richard Llewellyn from Bioresources. These presentations are about the amazing little Trichogramma wasp that we call MacTrix, an egg parasitoid of macadamia nutborer. Its widespread use in macadamias has enabled massive reductions in chemical use to control nutborer, once the most serious pest in macadamias, causing high crop loss as well as requiring costly monitoring and spray programs. MacTrix is now considered one of the most successful biocontrol agents in Australia. But we still get a lot of queries from growers, especially those new to the industry, who are unaware of how serious a pest nutborer can be and are unaware of the development and use of this biocontrol agent. So we thought it was timely to do these presentations. In part one, I'll talk about the development and the adoption of this wasp as a biocontrol agent, and a bit about the biology of macadamia nutborer and, of course, the wasps. In part two, I'll talk about the way the wasps are distributed in the field and their ordering and delivery process. And in part three, I'll go into more detail about monitoring for MacTrix parasitism. I've been working in the area of biological control since the early 1980s and worked developing a number of commercial biocontrol agents in that time. And in February 1998, I happened to be in a macadamia orchard near Alstonville with Alan Coates. I was new to macadamias, but quite familiar with Trichogramma wasps, having worked in deciduous fruits, grapevines and field crops, and knew the telltale signs of parasitism. We found a nutborer egg that to me looked parasitized. We found and collected more, and I hatched out the wasps and tried to rear them on light brown apple moth eggs, but with very limited success. Gus Campbell, who was the DPI entomologist at Alstonville, had a lab culture of nutborer that was mainly used for chemical assays. Alan Coates was able to collect more parasitized eggs and get them to Gus, who was able to get a small culture going on nutborer eggs. As it turned out, the wasps were a species that is thought to have entered Australia from Africa in the previous decade or so. There are a few other sightings on the Sunshine Coast. Its correct name is Trichogramma cryptophlebiae, so it's not surprising that we just call it MacTrix. Its host in Africa is a moth species almost identical to our nutborer. Bioresources and the New South Wales DPI then began a joint HAL research project where Bioresources would focus on developing the larger scale mass rearing system to enable us to do more trial releases. To rear natural enemies in big numbers, you need lots of suitable hosts. So a lot of natural enemies are mass reared on alternative hosts that may be easier to rear. Some other Trichogramma species are reared on easy to produce grain moth eggs, but we found the MacTrix wasps would not parasitize them. MacTrix, we found, had a very, very narrow host range and much preferred nutborer eggs. So we focused on developing an efficient system for rearing nutborer. We had to greatly modify and upscale the lab culture method and develop ways of handling large numbers of moths and being able to collect their eggs. This involves designing and building equipment that takes into consideration the development and behavior of the moth eggs, the caterpillars, the moth adults, and of course the wasps. We had some ups and downs, but eventually developed a reasonably efficient system. In other crops, Trichogramma are often released in very high numbers, hundreds of thousands per hectare, but in our case this was not going to be practical for the reasons I've just mentioned. With experience of other biocontrol agents, we settled on a dribble technique as the most likely to be successful, as well as capable of supplying many growers in a season. We did some small in-tree trials which showed the wasps were good at finding nutborer eggs, but needed to test it on a wider scale. Once we were able to get a reliable culture going, as part of the research project, we decided the best way to test it out was to get them to as many consultants as practical to trial in small plots. This hands-on approach proved to be very helpful to consultants who quickly learnt to identify the telltale signs of parasitism and started to get confidence in the parasitoid's abilities. It wasn't long before consultants were reporting back with observations of high levels of parasitism in areas where nutborer was a real problem, and were making don't need to spray decisions. This feedback process was an important part of the early stages of the annual Macadamia Consultants meeting. Since that time, the use of MacTrix really took off, and soon many growers were releasing wasps each season. There were clear successes within farms, but we were still a little surprised after several seasons to realise that nutborer levels had dropped through whole districts as the wasps had spread to farms that had not released them. Many farmers did not realise why their nutborer numbers had dropped. Nutborer remains in the environment, more so in some areas and farms than others. Damaging levels of nutborer are still found on isolated farms or pockets where MacTrix has not been released regularly. It's clear that without the presence of the wasp, nutborer would return to its previous major pest status. There has been a resurgence of nutborer on some farms where regular spraying for Sargastus has made it difficult for MacTrix to reach its potential. Macadamia nutborer moths lay their scale-like eggs directly onto or near the nuts. It takes about 4 or 5 days for the larvae to hatch, then they bore straight into the husk. If the shell is still soft, they can bore into the kernel, and if it's hard, the damage to the husk will still cause premature nut drop. In summer, from moth egg to adult takes about 5 weeks. If a larva is able to develop and pupate in the nut, then the adult moth emerges from the pupal case, which remains stuck in the nut. The short development time and exposure of the egg and the cryptic nature of the larvae make this pest a hard one to target with chemicals, especially when in the tops of tall trees. MacTrix wasps can parasitize nutborer eggs that are up to 3 days old. By parasitize, we mean the females lay their eggs into the nutborer eggs. They may lay 2 or 3 into one nutborer egg. The wasp inspects the egg and sizes it up to see how many eggs it can lay. It also picks up the scent of previous wasps that may have already parasitized the egg. The wasp egg hatches and the larvae eats out the contents of the nutborer egg, killing the nutborer larva in the process. The wasp larva develops inside the nutborer egg and goes black after around 5 days. It then develops through to the adult stage and cuts a small escape hole in the egg. So instead of the nutborer egg yielding a caterpillar, 2 or 3 wasps emerge instead. This process only takes about 9 days in summer, so the wasps can multiply really quickly if there are eggs in the crop. Once emerged, they head off to find a mate, and the females disperse looking for more nutborer eggs to parasitize. The wasps have very fine fringes on their wings, enabling them to drift on light breezes. They are often called aerial plankton. The adults don't need much food, but feed on plant sugars and nectar, and also honeydew from aphids or scale. Although the narrow host range of MacTrix has made it harder for us to mass rear them, it is perhaps this focus on nutborer eggs that makes it such a great biocontrol agent. It's able to search through large trees with dense foliage to locate the small target that is a nutborer egg. This is certainly a much more efficient process then cutting and spraying thousands of litres of insecticide in order to cover every single leaf and nut on the farm. We use what we call a regular release technique, sometimes called the dribble technique, where relatively small numbers of wasps are released each week for 8 to 12 weeks, and starting several weeks before nutborer are usually noticed. This general method has been used to great effect with lots of biocontrol agents around the world. It catches the early laid eggs that help to build the population quickly and aids early wasp dispersal through the crop. A lot of biocontrol happens at a very low, barely detectable level, and often standard monitoring methods do not pick up the very low numbers of pests that may be on the farm. For instance, if you're looking for nutborer and look at 100 nuts, you may not pick up the one egg in the 500 nuts, but this may be equal to 1,000 eggs per hectare. And these early laid eggs are a resource on which to start the MacTrix population and to help them get dispersed through the crop. We call this a preemptive or proactive program, not reactive. MacTrix does not overwinter very well in most districts, as it's too cool for nutborer moths to be active and laying the eggs that the wasps need to keep them going. So releases ensure they get going in your crop early in the season. Starting to control a full-blown nutborer infestation in January is difficult. So releasing earlier, if you can, means that the occasional spray or bad weather event, whether extreme heat or wet, does not destroy your wasp population. They should then recover and releases can continue. The whole strategy is a type of risk management, as we don't know in advance when an adverse event may occur, and similarly, some releases will be more effective or timely than others, and it's hard to know this in advance. It's best to consider that you are ordering and paying for the whole program, not individual releases. It's generally agreed that biological control of a pest is the best option when it's available. But biological programs need not just be considered as a once-off release by a government agency called classical biological control, where the results and the distribution of the biocontrol agent can be very patchy. Beneficials need a supply of food and generally get through the cooler months in very low numbers. So relying on them to build up in spring is fraught with uncertainty. It may or may not happen. MacTrix has shown how regular releases of a good biocontrol agent supported by a commercial insect producer can have widespread impact. And having good biocontrol agents is an added incentive to adopt IPM. MacTrix use is effectively an area-wide management program maintained on a commercial basis. That is, one paid for by the growers, not a government agency. It's a great example for managing pests in macadamias, as well as in other industries. And it's a model that needs to be adopted and supported by growers and ag departments if we are to sustainably manage pests going into the future. But as I've outlined, it's not easy getting these programs going, as they require significant commitment from government, researchers, growers, consultants, and commercial insect producers to find ways to make them work. There are risks associated with it, and it's rarely clear early on how this process will develop. However, when it does come off, it's a solution for the foreseeable future, not just a short-term fix.